My name is Ashley Mulford, and today I will be presenting on multi-omic association studies identify novel genes and proteins regulating cellular sensitivity to chemotherapy in diverse populations. So one important thing to remember throughout this presentation is that our DNA is 99.9% the same. Of course, as geneticists, we are interested in the variation that does exist, as that's what goes into regulating the phenotypes that we're studying. However, it's still important to remember that genetically we are much more similar than we are different. So that being said, for this project, we are studying chemotherapy drug response, and we wanted to take a multi-omics approach to the association studies that we were conducting. So we looked at 3 different traits. In this approach that we took, we looked at genomic, transcriptomic, and proteomic traits. So we looked at DNA, RNA, and protein levels in order to get a more complete picture of what goes into regulating the chemotherapy drug response phenotypes that we studied. And then we also wanted to study diverse populations in this project, as different global populations have different SNPs and genetic variants within those populations that are unique to both their continent and the individual population. So we looked at a Yoruba population, a Central European population, and an East Asian population comprised of Chinese and Japanese individuals. And genetic research also just has a lack of diversity, with the vast majority of studies being conducted in European populations. So we also want to try and bridge this gap and include more diverse populations in our genetic research. So as I stated, our initial data came from these 3 1,000 genomes populations. We had genotype dosages for our YRI, CEU, and ASN populations that we used, and then we also looked at these 3 populations together in an ALL combined cohort. The YRI and CEU populations contain parent-child trios, so we accounted for relatedness when conducting our studies, and then we also accounted population stratification when looking at our combined cohort ALL. And then for our phenotype data, we were looking at drug response phenotypes in the form of IC50 and AUC measurements for a total of 8 different chemotherapeutic drugs. And these are just measures of cell viability after treatment with a corresponding chemotherapeutic. Using this data, we were able to conduct transcriptome-wide association studies with PredictScan and GEMMA. So for PredictScan, our input is the genotype data from these 3 populations, and then our RNA prediction models that were previously generated and can be used to predict gene expression levels. So we ran PredictScan to generate this predicted transcript levels, and then we used GEMMA to find associations between our predicted transcript levels and our chemotherapy drug response phenotypes. And after running GEMMA, we were able to identify significant gene transcripts. So our top hits from TWAS with PredictScan and GEMMA, we found 1 gene significantly associated with etoposide drug response. That would be STRD5, and then we also identified a gene significantly associated with capecitabine drug response, and that's USF1. And so for STRD5, that's a star-related lipid transfer domain, and for USF1, that's an upstream transcriptome factor. And we wanted to look further into our most significant hit, STRD5. So when looking at this, we determined that as STRD5 expression increases, the concentration of etoposide needed for cytotoxicity decreases. So we just plotted the predicted gene expression against our etoposide IC50 phenotype measurements, and we found that at higher levels of predicted gene expression, there is a reduced need for etoposide in order to achieve the same amount of cytotoxicity. So when STRD5 is more highly expressed, etoposide is more effective, there's higher efficacy there, and the cell lines are more sensitive to this drug. So we wanted to then conduct functional experiments for STRD5 to try and validate this association that we identified computationally. So we performed gene knockdown experiments in an A549 lung cancer cell line, which we then treated with etoposide. And we chose this specific cell line because etoposide is commonly used to treat lung cancer. So the way that these experiments worked was that we performed gene knockdown using siRNA for STRD5 24 hours prior to treatment with etoposide, and then etoposide treatment occurred over the course of 96 hours. And we also measured the relative STRD5 expression at each of these time points, so at 0, 72, and 96 hours, to confirm that gene knockdown was consistent across the entire duration of the experiment. And then we were able to validate the results that we had predicted computationally through these experiments, as we found that the knockdown group, which is represented in purple, had significantly higher relative viability than the control group represented in orange on these plots. So you can see that at both 72 and 96 hours, with increasing concentrations of etoposide, relative viability was significantly lower in our control group with higher STRD5 expression than in our experimental gene knockdown group. So this validates our predictions and the association that we found that demonstrated that as STRD5 expression levels increase, the cellular sensitivity to etoposide would also increase. So in addition to conducting transcriptome-wide association studies, we also wanted to conduct proteome-wide association studies. However, there were not previously generated prediction models for proteins as there were for RNA. So we went about making these prediction models ourselves using data from the TopMed Consortium and the MESA cohort. So this data was proteomic data that had been collected from blood plasma from 4 different populations, so an African American, European American, Chinese American, and Hispanic American population. And the way that this was that protein aptamers were used to measure these protein levels in these 4 different populations. So they used SomaScan aptamer-based assays for a little over 1,300 different proteins, and these are DNA that had been modified with amino acid R groups to be able to bind to proteins and then measure protein levels, which we were able to then associate with the genomic data that we had from these populations so that we could include significant SNPs associated with differing protein levels in the prediction models that we generated. So the prediction models that we built were built using ElasticNet. They were trained in the 5 different populations, so each of the 4 populations separately, and then an ALL MESA population with all 4 of these MESA populations together. And these prediction models included significant SNPs only within a 1 megabase window to be used as predictors for protein levels. And we also used an alpha mixing parameter of 0.5 and a Spearman correlation of greater than 0.1 between predicted and observed levels as a determination for which models would be considered significant or not. And then all of these models were formatted with use for PredictScan, so we could conduct our association studies using the same PredictScan to GEMMA pipeline. So our Pwas results, we identified 7 unique proteins associated with chemotherapy drug response for 4 of our different chemotherapeutic drugs. And for the most significant, we identified the protein N-acetylglucosamine kinase, which is encoded by the gene NAGK. And we found that as protein levels increase for NAG, K, N-acetylglucosamine kinase, cisplatin IC50 also increases. And so this protein is known to regulate the Wnt signaling pathway, which is a pathway that's known to be associated with cancer as it controls metabolism, cell growth, and cell proliferation. And so it can sort of make sense that as you'd have increased protein levels for this protein, if it's upregulating this pathway and it's promoting cell growth and proliferation, it would make sense that you'd need a higher concentration of your chemotherapeutic drug to result in cellular sensitivity and cytotoxicity. So in conclusion, we identified associations between multi-omics traits and chemotherapy-induced cytotoxicity across our diverse populations, and we identified 2 significant genes and 7 significant proteins. And we were also able to validate our most significant gene hit, STRD5, using gene knockdown experiments. So we were able to demonstrate that what we found computationally was able to be replicated in these knockdown experiments. And so for some future directions, of course, all of these associations were identified computationally, and we were only able to follow up on one of them. So further studies would be needed to fully understand all of the functional implications of these findings. But overall, we are making strides towards more efficient treatments. And this, of course, requires diversity in the traits and in the populations that are being studied. All right, so now I'd just like to acknowledge the people who supported and contributed to this project. And at this point, that concludes my presentation. Thank you all for listening.